This is a study looking at a very specific population of KRAS mutated patients. We know that KRAS is a very difficult issue in terms of our non-small cell lung cancer and other cancers, but specifically for non-small cell lung cancer. So this is a compound that inhibits SHP2 or SHP2, which is a phosphatase enzyme. And a very significant study is that observations were recorded in terms of therapeutic responses. This is a phase 1b study and looking at various dosages. And this was a classic 3+3 type of a design. The study found that indeed this was a very tolerated regimen, and the grade 3 and grade 4 toxicities were not that much. And clearly it is in response to KRAS inhibition. And so what the study really reported is that there are T cell infiltration within the KRAS mutated tumor samples, which is important. And they saw T helper cells as well as cytotoxic T cells. That's one of the first significant findings. Another significant finding was that the intermittent dose was quite effective. The third significant finding was that these KRAS mutated patients did respond, and you had overall response that was significant. At the same time, what was interesting to me was that there were KRAS responders to not only G12C, but other codon 12 mutations for KRAS mutated non-small cell lung cancers. So RAS is a very difficult oncogene to treat, and we've been working with this for over 30 years now, trying to inhibit RAS. And there have been many strategies, including in the past farnesyltransferase inhibition, gene therapy types of therapies, but KRAS G12C potentially has been broken in terms of its code. And there are two drugs that are very important, that is sotorasib and adagrasib, and hopefully they'll get approved by the FDA over time. But we also know that sotorasib and adagrasib don't necessarily have an indefinite progression-free survival or an overall survival. So we have to be able to determine how we can inhibit KRAS for much longer PFS as well as an OS. So SHP2 seems to be a very important downstream target of KRAS, and it is important to inhibit SHP2 or SHP2. And what the authors showed here is that they can inhibit it with good significance and without that much toxicity. So for the future, I'm looking forward to combinations of KRAS inhibition, such as G12C, in combination with the anti-SHP2, or even combining anti-SHP2 with immunotherapy or other downstream signaling events. We're excited for our non-small cell lung cancer patients. You know, I've been practicing medical oncology for over 30 years, and I'm a thoracic oncologist, and we really are seeing the light at the end of the tunnel. Certainly, we've seen it for EGFR mutated patients or ALK translocations or RS1 translocation, exon 14, and it is important to know that KRAS, which we thought we could not break the code, so hopefully the code is being broken.